everyone. Today my topic is the structure of striated muscle. So before discussing about the structure of striated muscle, we are discussing about what are striated muscles. So first of all, it's muscles. The human muscle constitute about 40 to 50% of the human total body weight. Now the types of muscles. The muscles are classified into mainly three types, that are striated. The second one is smooth, and the third one is cardiac. The striated muscles are also known as voluntary muscles because why they are known as voluntary muscles? Because they are under the control of the animal's will. The smooth muscles are known as involuntary because they are not under the control of the animal's will, and the cardiac muscle, as the name originates, it means the heart. The muscle of hearts are known as cardiac muscle. Now striated muscle, where they are present. The striated muscles are found in limbs, in body wall, in tongue, in pharynx, and in the beginning of esophagus. Whereas the smooth muscles, they are present in the posterior part of esophagus, in stomach, in intestine, in lungs, iris, and dermis. Where the iris is present, iris is in the eye and dermis of skin. The cardiac muscle, as I have already discussed, it's present in the heart. These are the muscles of heart, and so they are found in the wall of heart and large veins. So what are large veins? The large veins are pulmonary vein and superior vena cava. Right.
Oi.
Now the striated muscles have myoglobin. What's myoglobin? The myoglobin is the oxygen carrying pigment that's present in the muscles, and the striated muscles contain ATP, urea, and creatine. Creatine is the amino acid. And the smooth muscles, they are never connected to the skeleton. I forgot to mention one thing, is that they are under control of animal's will, whereas the smooth muscle, they are not under the control of the animal's will. In turn, they are controlled by autonomous nervous system. Whereas the cardiac muscles, they show the character of both striated as well as non-striated muscles. Smooth muscles show characters of both the muscles. Now we proceed the detailed structure of the skeletal muscle. Detailed structure of skeletal muscle. Now suppose this is a muscle of limb, of arm, we can say. These are the muscle and these are connected with the tendons. Now the muscles are made up of the muscle bundle or fascicles. So if we take the section of the muscle, each muscle contains the muscle fiber. This is the muscle fiber. This is complete the muscle bundle and it contains the muscle fiber. Though the single muscle fiber is known as muscle cell, each muscle fiber is surrounded by the membrane called sarcolemma. This is the plasma membrane. Suppose this is the muscle. This is sarcolemma. This is the plasma membrane of the muscle called sarcolemma. The cytoplasm, this is the cytoplasm. It contains various nuclei. The cytoplasm of the muscle is known as sarcoplasm, as it contains the multiple nuclei. So this muscle fiber are known as syncytium. That means it contains the multiple nuclei. Now if we contain this muscle fiber, this we contain. So this muscle fiber contain number of myofilament or myofibril. This is the myofibril. The muscle fibril that contain various number of myofibril. Each myofibril contain dark band that are known as A band. These red color patches, they are known as dark band, and the light color bands are known as light I band. So now this is the myofibril. The myofibril contain A band that are dark color band, the I band that are light bands, the axon that is present between the A band, and this is the Z line and this is M line. So the myofibril here distinguished into two types, A band and I band. Now the myofibril contain two types of bands, that are A band and the second one is I band. The A band are the dark color bands and are known as isotropic bands, and whereas the I band are the light color bands and are known as anisotropic bands. Now the center of the A band, this is complete the A band. Now its center is known as axon or Hansen zone. This is axon. The center of the I band, where the I band, this is I band. The center of I band contain the Z line. This is Z line. It's the zigzag Z shape. This is Z line. So the Z line, they are the cross membrane or Dobbe's line. They are also known as Dobbe's line. Now the center of axon, that is known as M line or middle line. This is middle line. Now if we take the section of this muscle, it contains the thick filament, the thin filament, and these are the cross bridges. Now we will discuss about sarcomere. So what's sarcomere? The part of myofibril between two successive Z line, that is known as sarcomere. The part of myofibril between two successive Z line, they are known as sarcomere. The sarcomere contain A band, I band, this A band, I band, and this is Z line, and thick filament, this thin filament. The thick filament, size of thick filament is 150 angstrom and the thin filament size is 70 angstrom. Now how the thick filaments and the thin filament, they are combined with each other. We'll discuss they are combined with the protein that we are going to discuss about. Now initially we are going to discuss about the thick filament. The thick filament consists of this myosin protein. The protein chain is contained. The thick filament contain of the myosin protein. This is myosin protein, and this is myosin head and myosin tail. The myosin head consists of the globular region. The myosin protein that's split up by the enzyme trypsin into two fragments. The two fragments are light meromyosin and another one is heavy meromyosin. The light meromyosin that lacks ATPase activity that lacks ATPase activity and does not combine with the actin. And does not combine with actin. Whereas heavy meromycin that contain the ATPase activity and they combine with the actin like this. This is light meromycin and this is heavy meromycin. The light meromycin does not contain ATPase activity that bind with the actin. This is actin that we will further discuss. This is actin. So these are combined. This does not light meromycin. They do not combine with the actin, whereas the heavy meromycin they combine with the actin. So now we are going to discuss about the thin filament which contain the actin. So now what is thin filament? Thin myofilament. The thin myofilament composed of 3 proteins that are actin. The second is tropomyosin and the another is the troponin. The actin has low molecular weight. They occur in the 2 forms, monomeric. Monomeric form is also known as G-actin and another one is polymeric. The polymeric form is known as F-actin. Like this, these are the actins. If it is present like this individually, they are known as G-actin. If it is present in a bundle, then this is F-actin. Now tropomyosin. The tropomyosin is made up of double-stranded alpha helical rod, and the tropomyosin is attached with the F-actin. F-actin, this complete is F-actin. This is tropomyosin, this brown color. The tropomyosin is attached with the F-actin. And the last but not the least, the troponin. Troponin is a complex of 3 polypeptide chain. The first is TPT, troponin T, that bind to the tropomyosin and other 2 troponin component. The second is TPI, that inhibit the F-actin myosin interaction. That it does not allow the troponin to combine with the F-actin. And the last one is TPC, that contain the calcium binding polypeptide chain. This green color, they are the actin. They have low molecular weight and are categorized into monomeric and polymeric form. The monomeric form are known as G-actin and the polymeric form like this, they are known as F-actin. The another is tropomyosin. It is made up of double-stranded alpha helical rod, this brown colored. They are attached with the F-actin. And the last is troponin. They contain the 3 polypeptides. The first one is TPT, that binds to the tropomyosin. It binds with the tropomyosin here. And another 2 troponin component. The second one is TPI, that inhibit the F-actin myosin interaction. Like here we can say it's present. It does not bind with F-actin. Does not bind here. So then last one is TPC. Above it binds with the calcium. So thank you so much. If you liked my video, then share it with your friends. So guys, keep studying. Bye bye.